Hello and welcome to Health Cities Talks. In this edition, we're going to be talking about the Bio-Conversion Databank Foundation, which is working to make microbiological knowledge easier to access, more efficient to research, and easy to apply to any industry. My name is Mac Mail, and I'm the co-founder of Taproot Publishing. Every Wednesday morning, we publish the Health Innovation Roundup, sponsored by Health Cities, which is a curated email newsletter that brings together the latest headlines and happenings in Edmonton's vibrant health innovation sector. My job is to learn about all of these interesting things going on, and I'm excited to learn more about bioconversion today. I'll try to keep us on time and shepherd the conversation along, but let's dive in. We have two guests today. First is John McDougall, who is the CEO of Bio-Conversion. Welcome, John.
Thanks, Mark.
And Dominic Sauvageau, who is the engineering biology lead at Bio-Conversion. Welcome.
Thank you.
All right, I want to start with a little bit of context. And John, I'm going to ask you a little bit about that. So some people like to remind us that we're in this fourth industrial revolution. We've got artificial intelligence and big data and everything that goes along with that. But not always do we think about biology. But I understand that context is really important to how bioconversion came about. So can you give us the lay of the land here?
Well, that's a great question, Mac, because certainly we're in a transition. We can all feel it in this province and everywhere else that we go and listen and look. It is often called the fourth industrial revolution, and it is built around certainly data and artificial intelligence and the Internet of Things and so on. And as you point out, genetics is a big part of it. It's a big part of it because, of course, it enables a transition to a much more circular economy, which seems to be kind of where we're going. So it takes all of these things, builds them together, and with DNA sequencing, with the ability to do analytics against massive data sets, which is what biology is all about, with the ability to apply machine learning and so on, we can now get into a space and, in a sense, almost design the future through biology, messing around with organisms at their most fundamental levels.
I think it's pretty incredible that CRISPR, it seems like, has become almost a household term. Like lots of people know what that is, which is fascinating. You mentioned the circular economy there briefly. Do you want to expand on that a little bit? What do you mean by that, and how does that factor in?
Well, the circular economy is really a polite way of talking about biomimicry. It's really trying to think the way biology acts, if I could put it that way, and put it into practice. So things that emerge as waste in one application are used as inputs in another. And in a sense, the whole system is in a constantly mutually reinforcing process. So we don't end up with the big problems like we do today, where we harvest long stored things and then throw away the waste and then do it again and so on.
So a big source of waste these days that is on everyone's mind is greenhouse gases. And I understand that had something to do with how bioconversion was born. Can you tell us a bit about the origin story for the foundation?
I can. The whole thing actually began by looking at the challenges of climate change and greenhouse gas emissions, how they were growing and so on, and recognizing that most solutions were, I would say, not terribly value-adding solutions. Most of the time it was, how can we hide it away, put it in the closet, bury it in the ground, do something and forget about it. It came to our mind that actually, in essence, we've got factories all over the world producing a product which isn't being used. And if we could use it, then and add value to it, then we would have our hands on something pretty special. It would solve the problem and it would also generate wealth. So we started down the path of the Bio-Conversion Foundation from that perspective.
And Dominic, I understand one of the things you started doing was looking for organisms maybe that took greenhouse gases, carbon emissions as input. And I imagine one of the ways you'd go about doing that is data sets. There's data sets out there of organisms, but there's a lot of challenges with those, right? Can you help us understand what are some of those challenges with data that you very quickly ran into and that you're now trying to address?
Right. So a number of people and groups around the world are working with microorganisms that are able to take different greenhouse gases and transform them in one way or another. So it could be CO2, it could be methane, and others. So again, that's a very important element in combating climate change and has a lot of potential. But one of the things is that these organisms are not typical, well-known organisms that are used in a lot of technologies. First of all, they're not typically health related, which means that we often have less information about them. But here's the thing, that at the same time that we're trying to develop technologies on this side, there's also a huge amount of data that's created on the environmental side, which is things like metagenomics, where the genetic material in a whole environmental samples is being analyzed and people are reconfiguring it to try to investigate which organisms are there. So this is extremely valuable in terms of information, but it's not always tied directly in terms of us understanding exactly which organisms are there and how they're using the different functions that we observe in the data sets. So it creates these data sets that are abundant, but they're not necessarily very clearly defined. And because these organisms are not always very well characterized and used by thousands and tens of thousands of researchers around the world, the information is also not complete and also sometimes erroneous. So there's a real challenge there. There's an opportunity and a challenge at the same time in tying the information to actually something that we can create new technologies with.
So I think I'm hearing kind of two big problems, maybe more, but the first is that the data itself is maybe not very clear or well annotated, or maybe even consistent. And so that's one of them. And then the other is, it sounds a little bit like it might be siloed, right? So if you're working in environmental studies versus you're working in health, that data isn't always brought together. Is that a correct assessment?
Yeah, I'd say so. And it's siloed in different ways. There's a lot of effort to try to make this data as available as possible. So the data is often made public, so it's freely available for people to consult. But again, we, as humans, we often create these categories of applications. And then, as you said, we may think of health as one thing and as environmental information as a different thing, but they can be related. And the other way also is that it's not trivial to go and retrieve the data and be able to interrogate it properly, to analyze it, right? So there are some technological challenges. There's specific training that's required often to be able to access the data and to get the information out. And that can take a lot of time and resources that are not available to all.
And then once you've got the data, you want to do something with it, presumably. And maybe before we talk a bit more about what the Maximizer platform from Bio-Conversion does, maybe John, you could help us understand what is the traditional process here? So you've got this data, somehow you've got some expertise in that data, you've figured out that maybe you've got a list of candidates. It sounds like there's a lot of manual work involved in that step, but there's even more, right? And it takes a long time. Can you help us understand what, like before Bio-Conversion came along, what typically happens?
Well, typically, and Dom is more familiar in this than I am, but I'll start and pass it over to him. First of all, you would go to somewhere that had data. There's sites all over the place that have this data. And the first challenge is the structure and just even finding your way around their site, right? So they've gobbled up lots of information, but they're not necessarily providing it consistently. Their sites are not necessarily intuitive in terms of how you work around them. So you almost need an expert in order to be able to even use the site.
Right.
So the first challenge is find the data, get it and extract it, bring it out in a form that in fact allows it to be worked with. And then you can start the annotation process to try to make it consistent and usable and comparable. But I'm going to pass it from that point over to Dom.
So you got the data, now you're into trial and error, I imagine?
Not quite, but.
Not yet. Not yet. We do get the trial and error eventually. But yeah, once you got the data, that's where, and I don't want to minimize, there's been extremely great effort that's been put in trying to find ways to facilitate the access to the data and being able to read it and everything. But it remains that, as John was saying, there's a lot of skills and a lot of expertise that needs to be developed to be able to do that. So often, every time that a company or research group has an idea, they have to go and retrieve the data, treat it in a way, and before they can even start to think about integrating the information in their technologies. And that is replicated for every group and every company and every potential user of the information, which means that the interface with the data is much further than what we would hope it would be, right? So there's a lot of work that needs to be done. Then once you have that data, once you have that information, assuming it is good, then you can implement whatever technology you have. So if you have the idea of using a gene for a certain application, you go retrieve the information on that gene, maybe the sequence, and you build your technology around it. And then this is where the trial and error comes in. Whereas if the information is very good and your idea is well executed, you may be lucky and get it the first time around. But what happens way more often is that we have this iteration process where we don't fully understand the full system. So we have to go and tweak and change a little bit our strategies. Now, the problem comes when the data that you retrieve, you think that a certain gene has a given function, but it's the wrong function. And this is where that still happens too often in health, in environment, in bioproduction, bioconversion, that the fact that our access to the information is automated in a way that sometimes mistakes come in, and it means longer, more expensive development of technologies.
And so, that leads us, Mac, that then leads us to when you actually start to work with it, right? And then you have to be able to do the data analytics. And again, the challenge is that kind of work also took experts and expertise. They had to really know their way around the genome and biology and the data, and it would take them, often months or even years to try to put these things together.
But it doesn't have to, right? It can be a lot quicker, and this is where the platform comes in. So I've seen a brief demo of the Maximizer platform. Dominic, you were showing me. It's very visual, even for somebody like myself who doesn't know anything about the underlying data. I can see how it's used to navigate around and how powerful it can be to explore that information. So it sounds like a big focus of that platform has been on the data side, the curation of the data, making sure that it can be displayed in such a rich visual way. Do you want to talk a little bit about that? And then what is the next step? What the data is used for some purpose, presumably. And as we've already highlighted, machine learning seems to be the thing that loves data these days. So can you walk us through a little bit how somebody would use the platform in that way?
That's right. So to put it in context, you're absolutely right. So, the role of the BioDF platform, the Maximizer, is to be able to retrieve the data, curate it, and make it available in a way that's very instinctive, very visual, very easy to manipulate, and then be able to have access to the full level of the data. So for an expert that comes into the platform, it becomes very easy to access the data, to tap into it, and then to very deeply into, for example, sequences of a gene or something like that. For someone who's less well-versed or who has different needs or needs to set up a new technology on a very conceptual level, then you can have the information available in a very visual, qualitative, in a very visual and qualitative way. So that allows the exchange between people who have different levels of expertise to be much better than what's currently available in a lot of forums. So it really creates a workspace for all that where people are able to exchange concepts and ideas on new technologies, data, and everything, but in a way that's very commonplace. So it creates kind of a new language for people to talk about. So how does AI come into this? Is for us, we have to retrieve this data and treat it in a way that's very efficient and fast with a high level of automation. So this is where we can use AI to retrieve the data efficiently. That's the first one. So that's one aspect. And then the other one is on the curation and validation of the actual accuracy of the data. So this is where different machine learning strategies are used to actually treat the data and for us to be able to inform on the quality of the treatment.
At the end of the day, if I could, just for a second,
absolutely.
the challenge that that the user ultimately is dealing with, is they want to come up with a design for an organism, which might be a modification of a current one or the development of a new one, that will actually carry out a certain function with a degree of efficacy.
Right.
And so they're trying to drive through, and they have to be able to reach into the data, find things that may help them down that path, find places where they have near complete capability that might be supplemented by some near neighbor who has not all those capabilities, but does have one of the missing ones that might be important, and so on, so that you can end up basically building what amounts to your own design for your biological process.
Yeah, and that's where the third aspect exactly of the AI tools is on the prediction, right? Where we aggregate, we curate, we give good data, but then once you have good data, you're also able to do an extra level of analysis and possibly potentially predict the behavior of an organism, predict a more efficient sequence for a gene, et cetera, et cetera.
So this is really interesting because that's where that large time scale that you talked about, how it can take years or months, I guess, really becomes compressed, or can be maybe not compressed, but accelerated, right? Because you can do these models digitally. This is the same impact we're seeing machine learning and artificial intelligence have in all kinds of industries, right? Things that used to have to be done in a very mechanical, manual way can now be done using data to drive those outcomes. So that's really interesting. And the other thing, I think users of the system, they can upload their own data as well, right? In addition to the data that you're curating.
Yes, that's right. And that's where a big part of the BioDF platform is actually in facilitating the engagement of communities. Right. So the users are very much invited to provide their own information to help in the validation of the information as well. So really to be fully engaged in the evolution of the platform and the use of the platform. Yeah.
And John, I imagine that opens up some really interesting cross-industry, cross-fertilization opportunities. You guys started with greenhouse gases, as you mentioned, and now obviously it's being used for health as well. What are some of the other potential opportunities you see for the platform?
Well, you've got to be really careful. One of our challenges is not to try to go in too many directions at once, of course, but it is a very ubiquitous capability. So we started with greenhouse gases. We had early interest from people working in waste management and anaerobic digestion. We then sort of slid over to health in terms of areas of application that look quite exciting there. It could be used for many environmental things. It could be used for reclamation. It could be used for improving food production. I mean, the challenge here is not to try to enunciate all the possibilities, but rather to focus on a few and really drive them to fruition.
So Dominic, we've got a bit of an understanding about the platform now, about the curated data, the ability to do some modeling with that data, the collaborative aspect of it. Can you give us a use case? What is a typical use case from start to finish that you envision somebody following using the platform?
Right, and this is one of many potential examples. But if we're talking about health, and the initial focus of the platform is on prokaryotes or microbes. So in health, we're seeing more and more the rise of antibiotic resistance being a problem, and certainly. One example is a case where a patient in a hospital develops a bacterial infection that is resistant to antibiotics. And through the characterization, an MD can do a sample and then send it over to an analytical lab where there's going to be a characterization of the bacteria. And sometimes the bacteria doesn't fully check all the boxes of what we know so far. So it can be a variant of something that is out there. Usually this process is very linear and fairly long, but through the platform, what we can have is once the characterization of the bacteria is done, that information can be integrated into the platform, and very quickly and visually comparisons to other known bacteria can be done. And then the discussion can start between the MD and the lab into trying to elucidate potential mechanisms that are explaining the antibiotic resistance, and then can start to create potential targets for solutions. And again, this discussion can be done through the platform in a way that people can kind of find a common ground in their language. then this is where industry can come in, where a company can start to enter the discussion, and then again have a discussion at all levels with the MD, the analytical lab, and then the company, and they can decide to go after developing a technology or a target medication for that specific strain of bacteria. And that process, once you develop a train like this, where all levels of life sciences or the medical field are interacting and discussing, you can create a train where you're doing the development of these things much, much faster. So the first time around, you can take a few months before you start to have a potential solution. But as you're doing this, that can speed up much, much faster. And then creating two great outcomes, right? One is a patient that is healthy again,
right?
And in a way that's much more personalized. So personalized medicine is a big target here, but we can start to have these solutions that are developed really quickly for specific cases. And then the second one is that infrastructure that exists to speed up the process, creating these pipelines that can develop solutions in a much more rapid way. And I think I would argue that this could never really happen when we're having all of these players siloed and not communicating as easily. So although an MD may not be clear on the biochemistry of the sequence resulting from a certain gene, having that discussion and being able to talk about the impact on the physiology within a patient and talk about the biochemical impact on the mechanism of the infection can actually bring some great new solutions forward.
I mean, it sounds really appealing to have all of those different players connected, collaborating together, working in a common structure. There's probably lots of reasons why that doesn't typically happen or why it sounds so novel. People specialize, or maybe the tools and the infrastructure weren't there. John, how do you get all of those different players to use the platform? How do you get that kind of collaboration to happen?
Well, the beauty is, because the platform essentially is a virtual workplace, workspace, the people can stay where they are and they can still connect. So we've learned a lot about that through COVID and how that can actually be remarkably effective. So you can't simply pick up biofoundries and wet labs and companies and production facilities and so on and suddenly just bring them all together.
Right.
But this platform, in fact, does exactly that. It allows people to come in. It allows them to set up their communities and their groups in a way that's secure and private where they need to, so they can work quite confidently on their own area of interest and maintain their IP and so on. But it also gives them tools. I like to use an analogy. It's a bit like, say, the Microsoft toolbox, where you can get your PowerPoints and your spreadsheets and your word processing and so on. You sort of live on those platforms. Well, we're doing the same thing really with genetic data and the ability to process and analyze and communicate about it.
Dominic, I feel like we're talking about the platform like it's been around for years, but it's pretty new, I understand. Can you tell us a little bit about the uptake so far? Like, what kind of uses have you seen? What kind of data and stuff is available today in the platform?
Right. So again, to give a little bit of context of timeline, we're in September 2021, and this project, although it's been maturing for a while, really took flight in May 2021. So what we did is really start the infrastructure to enable access to data and the exchange of information and everything. So we're still like very early on, and yet we've been able to develop some great tools and great applications. The portal on the bacterial side is still about to be released, but what we've been doing is we've been developing a version of what's called the phage receptor database. Now, phages are viruses of bacteria that have many applications in health, in environment, and beyond. So again, this is one of those things where people like to tag one single application, one single sector for it, but it plays all over the place. And phages, in developing technologies that rely on phages, one of the main aspects is how do they recognize their hosts? So these viruses recognize specific bacteria. So the role of the receptor is very important. So we've been able to develop a version of the phage receptor database that is very visual, and again, with visual filters to facilitate the use of the database, access to the database, and use of information. So people have model bacteria with different organelles and also the components of the surface of bacteria, and they're able to click with this interactive visual and select the position, the type of receptors, the location of the receptors on the cells, perhaps even the type of bacterial hosts that are there. So by creating this, and we did a soft release about a month ago at a specialized conference on phages, and we already have over 50 users worldwide using the platform for different applications.
So and the response has been positive, it sounds like, from those initial users.
Yeah, very positive. And this is one of those things where we went from a static, database that looks like any spreadsheet to something that's very visual, interactive. Again, that the users can contribute to, because they can submit their own receptors to keep on populating this database.
Can you give us maybe a little teaser about what's coming up? What's the next big thing you're working on in the platform?
So the next big thing that we're coming up is the release again of the bacterial side of things. So we have these groups of bacteria that are related to both the environment and to health. One of them is methanotrophic bacteria that are able to convert methane into various products. And the other one is Clostridia, which are, again, if you think of Clostridium difficile, for example, is a common culprit in hospital-acquired infections. Some people will know Clostridium botulinum, because of two things. First, it's a very dangerous bacteria in many ways, but it's also led to Botox, which is an area that maybe not all users of Botox know, but we have these two groups of bacteria where we created structured databases, again, in our visual environment, where people can go and interrogate the whole genetics of these groups of 150 bacteria or so at once, and be able to do these comparisons, gain access to sequences, be able to build whole analytic projects and share it within the platform with other collaborators through different institutions. So all of that is just about to be released, and we're getting involved with different communities that are working with these organisms so that they can really start using it more and more. And that's kind of how the development of the platform goes. Once the infrastructure is built for these groups of organisms, we can use the infrastructure and implement it for different groups of organisms. So we can grow much and much faster this way, and be able to supply information on thousands and thousands of organisms for people.
As we do that, Mac, as we do that, Mac, we're in parallel bringing on more artificial intelligence and machine learning capabilities to constantly, if you like, simplify and accelerate the entire process. So what we've done over a few months, we'll probably do in a few weeks, and what we do in a few weeks, we'll end up doing in a few minutes, and so on. Right.
Well, John, it's interesting you mentioned that, because we're all located here in Edmonton, and we've got some pretty amazing institutions here that people probably have heard about: the University of Alberta and AMI, the Alberta Machine Intelligence Institute. There's some companies in this space like Drug Bank and others, and of course there's Health Cities, who's working to amplify all the transformational stuff that's going on. I'm curious, did any of those things make a difference in building bioconversion here? Is there either a really big benefit to being in Edmonton, Alberta, or maybe even a hindrance that you can see?
Well, I'd say the things that you mentioned are part of the reason that we are here, because the people that we need to work with wouldn't have been around otherwise. So that's very important. The challenge that we've got is our community tends to be more upstream than downstream. And what we need to do is make sure that we're popping out of what we do capabilities and ultimately enterprises that emerge from here and find it convenient and valuable to stay here, because they can work with organizations like you've talked about, but also, of course, bioconversion.
Yeah.
The other thing that I think is interesting is we've already got the nation now talking about this being a kind of a foundational piece for Canada's genomics and bioconversion strategies. So I think that that's going to help too.
Do you see a big role for Edmonton and for bioconversion in, I don't know, maybe the pharmaceutical industry? Like, what do you think is the big opportunity with what you're doing for this region?
The biggest issue is that it's easier to do things that where you have, I would call it, established industries to work with. Ideally ones that are pulling the entire value chain, not just trying to become maybe a small part of the value chain. So that's just a reality that we have to deal with. But I think we are at a very interesting time. I'll go back to the beginning of this conversation, and that is that we are in a time of transition, and that's true around the world. And so the people that kind of get out and into the race and sort of in front of the pack or near the front of the pack are going to have the best chance of really making it stick and stay.
Where do you see bioconversion going, John? Like, what do you see as the sort of next couple of years? What does the future look like for the organization?
What I see is building the communities that Dom had mentioned. So when we think of a particular area of focus, we have used the term community as a way to think about it, because it is a community, right? It's not just one or two, it's all the pieces of that particular ecosystem and trying to bring them together. So we've got the 2 or 3 that we're working on now. But the beauty of this, again, going back to the fact that it is so ubiquitous, with 3 or 4 people in bioconversion, we effectively can sustain and manage another community area. So the expectation is that these will steadily grow, and we'll get more and more. And established communities will probably, as they grow, evolve into niches, and then we'll have, if you like, sub-communities, and you'll have this thing just building on itself. The beauty is we've built a highly scalable capability. We can handle more users almost on the push of a button, and that's order of magnitude increases in use. So that's really important as well. So we've done everything we can, if you like, to try to position it as a really core element in the genetics and I'll say bioengineering space. And we've got some tricks up our sleeve in terms of how we apply computing power and a few other things that I think will help us stay there.
So Dominic, if somebody is watching or listening and they're quite interested to learn more, or they want to maybe get into the platform themselves, how does that process work? Like, do I just go and punch in an email and a password and sign up, or how do people get into the platform?
So the platform right now is the feature release are the FRED database, and it's that simple. It's really going to portal.bioconversion.org and just creating a profile, and then you have access to the platform. You've got your own profile, and then as the features are rolling out then they'll just become available. So the important thing is that the first interface of that platform is totally open. All people need to do is just create a profile and start using it. Now, if people want to talk about setting up collaborations and things like that, they're more than welcome to reach out to us. We do on the platform have the ability for people to provide some feedback and everything, or they can just contact us through our website as well.
Sounds great. You guys sound like you've done a lot with a very small team, small but mighty team. You've already made some significant progress. John, what other things are you looking for in terms of help and assistance? What else could help take, bioconversion from where it is today to where you want it to go?
The make or break of this is users. And so in that sense, what we really want and hope to attract is people who are, respected in their particular area, who get excited about what we're doing, who start to talk about it to others and sort of, in a sense, make the dominoes all starting to fall. That's really the key. I think that we'd love to see the strong health community here because, that's what has, in a sense, helped support the development of this platform in its first instance. We know there are other areas we can apply it to in the province. but I would really like to get some strong uptake through the health sector in Alberta.
Well, it's been very interesting learning more about bioconversion and the platform that you've built and that you're building. So thank you for sharing with everyone today. Is there anything that we haven't touched on that somebody should know about what you're up to, either Dominic or John?
You know, I think we've done a fairly decent job of covering, I think. But again, you know, the one thing we found as we were building and interacting with people is, you know, the community has good ideas. They can help us do what we're doing even better. And the more that they interact and suggest and propose, the better off we'll all be. Sounds
good. Well, thank you for sharing. If you want to learn more about the Bio-Conversion Databank Foundation, you can go to bio-conversion.org. John, Dominic, thanks again for answering all my questions, helping me understand this a little bit better. It's fascinating to learn about, and if you do get a chance to go look at the platform, I encourage you to do so. The visualness of it is pretty impressive, how powerful the tool is already at this stage. For more Health Cities talks, you can of course stay tuned to healthcities.ca. And if you're interested in what's going on in the Edmonton health innovation sector, I encourage you to check us out at taprootedmonton.ca, and you can check out our headlines and happenings every single Wednesday. Thank you all for joining us.
Thank you.
Thanks, Matt.